Hi everyone, my name is Amin, and I'm on the SIs for Dr. Maduro's BIO005A class, and today we'll be going over mitosis and meiosis. So to jump right in, I wanted to start out with explaining what diploid cells are and like what they mean exactly. So basically, our body cells, everything besides our germline cells are considered diploid. And essentially what happens is that one chromosome comes from mom, while the other one came from dad, right? So that's why we have 23 pairs of chromosomes, right? But technically we have 46 total chromosomes, right? Because one came from mom and one came from dad. So if we look at the next slide, you can see that over here that we have the sperm, right, which came from the dad, and the egg that came from the mom. So essentially you can think of it as 23 chromosomes came from that one and 23 chromosomes came from that one, which makes our zygote, which is what our body cells are made of, which has a total of 46, right? So essentially, if we want to make a bunch of sperm cells or eggs, then we're going to use a process called meiosis, right? Because I spelled that wrong, but that's because we're going from diploid to haploid, right? But if we're trying to go from 2N to more 2N, right? So like skin cells or just body cells that we're trying to reproduce, they're gonna keep our full genome of 46 chromosomes, right? One from our mom and one from our dad will always be present, and that's what we call those diploid 2N. So another way you can tell what 2N and N is, basically you can interchange the numbers. So not all organisms have 46 chromosomes like we do. So if something has 2N of equal to like 6 chromosomes, and a haploid cell would have 3. You just divide the 6 by 2, and so each gamete would have 3, n equals 3, right? So the cell cycle is really important in this case, right? So if we go through mitosis, we just produced a fresh cell, right? So say this is a cell for our eyes, right? Our eye cells probably rarely divide and are gonna go through a cell cycle arrest. So many cells don't just divide every time, or else that kind of creates cancer, right? Uncontrolled cell division. So that's why most cells like to go to arrest and just be what they are. Now, a lot of cells actually do divide, right? Like replenish. And so what you got to do is go through different phases. So G1 is when you divide cellular contents, right? So this is talking about maybe like organelles and stuff like that, not your DNA, right? So you divide all your organelles, making sure you have 2 of everything because you got to make 1 cell into 2 cells, right? And then you hit the synthesis phase right here. And that's where you divide specifically the chromosomes, the DNA, because you got to put 2 sets, right? One in each cell. And so that's done in the S phase. And finally, the last phase before you go to meiosis is called growth 2, so G2. And that's where the cell essentially double checks that I have everything, I'm good to go. If there's any repairs, they're made here. And basically you jump into meiosis after. And then you go through PMAT, right? Prophase, metaphase, anaphase, and telophase. And then telophase is usually combined with cytokinesis when we talk about mitosis and meiosis. Essentially, this is the cell cycle, and it'll help us explain mitosis and meiosis further. So I'd like to go through a little bit of vocabulary before I jump into mitosis. So essentially, we got to understand the difference between chromosomes, chromatids, homologous chromosomes, and whatnot, right? So in the beginning, we start out with the chromosome. Okay, this is before S phase. So this is like a normal cell cycle, probably like G1 you can take, okay, or even G0. All right, and this chromosome is also called a chromatid because it's also going to be ready to be replicated, right? And so in the S phase, you make the exact same version of this chromosome and attach it together with a centromere, right? And these are called homologous chromosomes. So homologous. All right, also known as joint sister chromatids, right? Because they're essentially the same thing. So that's why we refer to them as sisters attached through a centromere right here. What mitosis does is essentially split this apart and put it in 2 distinct cells, right? So we started with 1 chromosome in 1 cell. We replicated the chromosome and all its contents, right, in G1, S, and G2. And then once we had it ready to go, we formed two distinct cells, right? That look exactly the same though, right? Because they have the same exact DNA in mitosis, right? So if this started with 46 chromosomes, then we will end in 46 chromosomes in each of these cells, all right? So these are called daughter cells, and this would have been the parent cell over here. All right, so let's go through mitosis. Mitosis is fairly simple when talking about dividing diploid cells, right? So we start off with interphase, all right? So the easiest way to think of this right here is think of this one as your chromosome from your mom. And let's just say this one right here is our chromosome from our dad, right? Just as an example, we would have 46 of these instead, but right now, just to make clear, we're referring to these two specifically. All right, now it's a diploid cell. So our end products need to look exactly the same as this, all right? So in interphase, what's gonna happen is we're gonna start preparing for DNA, for cell division, right? So we're gonna condense our DNA, make it tightly wound, just condense it and get ready to go into prophase, right? So prophase is when chromosomes start to move apart and you start generating a spindle. So what generates a spindle is a structure called a centrosome. You can see right here. It's also right here, right? So these are essentially used to pull these chromosomes apart during cell division. So these these centrosomes form something called spindles, right? So in prophase, what happens is that the centromeres move apart. I keep saying centromeres. I meant centrosomes. Centrosomes move apart, right, and get ready to start dividing, right? And as you can see here, we have copies of our DNA, right? But these are exactly the same thing. So this is from our mom. So we made two exact chromosomes, right? And this one's from our dad. Now what's going to happen in metaphase is that these line up in the middle, right? Just so it's easier to divide. If they were in this structure, as you can see in prophase, then our spindle would not be able to attach correctly to each specific chromosome. So in the metaphase, we line them up in the middle, and the mitotic spindle attaches to each chromatid, and it basically starts pulling them apart in anaphase, right? Sister chromatids get pulled apart, and essentially now you have two cells ready to go, right? We have the chromosome from our mom, the chromosome from our dad, and then as we prepared in G1, we have like all our cell contents as well in each cell, right? Now we're ready to divide, and cytokinesis occurs, splits the cell apart, and we have essentially two cells that are identical to what we started with over here, right? So that's just a brief overview of mitosis. A big thing is you're starting with 2N and you're staying 2N, right? All right, so let's get into meiosis. It's a little complicated, so I want to start off with a little brief explanation. We're just gonna hover over. We're not gonna go in depth about like crossing over and all that. Just like a general description, real quick. So meiosis, you're taking a 2N cell and you're making it an N, right? So essentially taking it from diploid to haploid, right? So you're reducing the chromosome number by half, all right? So let's just talk about an X and Y cell, right? X and Y. So say you're a male, you have an X chromosome from your mom and a Y chromosome from your dad, right? So this is your diploid cell. Now, if you went through mitosis, you'll essentially just make another one of these, right? However, if you went to meiosis, you'd have a different story. So first step, we want to replicate our chromosomes, right? So essentially we'll have two X cells, or X chromosomes, and two Y chromosomes, right? We made a copy. And then we want to go through meiosis 1. So we're going to divide this up, right? So one of our cells will have the X's, and the other one will have our Y's, right? Sorry for the handwriting, by the way. All right, so this is meiosis 1, right? Now, I didn't draw out the full process. Obviously, there's going to be some crossing over and whatnot. That's what makes each human unique, right? We don't look exactly like our parents. And we'll get into that right after this slide. However, this is meiosis 1, right? And now we're going to go to meiosis II. And essentially, this is where we split up each chromosome again. So X goes here, the other X that we copied goes here, one Y goes here, and the other Y that we copied goes here, right? So as you can see, this is haploid. This is only N, right? Because a diploid cell has an X and a Y chromosome, right? Let's say like my cells, my normal somatic cells. However, my germline cells will have only either an X chromosome in it or just a Y chromosome, right? So essentially these have 46 total chromosomes, right? And this will only have a total of 23 chromosomes, right? N equals 20. Oh no. Because N equals 23. Sorry about that. All right. So that's just a key thing to remember. And that's why when we talked about having a sperm cell and an egg cell, so here's a sperm cell, right? And here's an egg cell, and that forms a zygote, right? So there's an N and an N forming a 2N, right? So one X, there can either go an X sperm cell and join with an X from the egg, and that'll make a female, or it can be a Y sperm cell, right? A male, and it can join with an X egg and make a male, right? So that's why they say you have a 50-50 chance of getting a male or a female, right? Because there's a 2 out of 4 chance of being X or a 2 out of 4 chance of being Y, right, in your sperm cell, and you'll have 1/2 probability, right? So this is a brief overview of meiosis. The main thing is to understand that we're going from a diploid to a haploid, right? And there's 2 phases, right? Meiosis I and meiosis II, and we'll get into that further right now. All right, so let's get into the details of meiosis, all right? The meiosis, as we said earlier, is split into 2 stages, meiosis I and meiosis II. Meiosis I is when homologous chromosomes separate, right? So essentially, let's look at this right here, right? This is already, it has already gone through interphase. Well, let's talk about what it will look like before interphase, right? So 2N equals 6, right? So essentially we start off with a diploid cell, 2N, right? And it has 6 chromosomes. So 1, 2, 3, 4, 5, 6, right? However, over here, we see that we have 1, 2, 3, 4, 5, 6, right? However, we see that there's 2 of them, right? And that's because they're sister chromatids, right? And that's because we went through interphase and made copies, right? So essentially, after interphase, it will look like this. So you have pairs, right? So 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11, 12. All right. Oh. So essentially, as you can see, we have a total of 12 chromatids after we have gone through interphase, okay? And so that's what we see here. Now, what's special, or a key thing to remember here, is that in this diagram, we're combining prophase. There's 2 prophases, early prophase 1 and late prophase 1. In this diagram, in our class, we're gonna combine them, all right? So just think of this as one step. And so, as you can see, in the early prophase, which is still considered just prophase, these non-sister chromatids will form basically homologous chromosomes will basically form a synapsis, which as you can see right here, and perform crossing over, right? This is very, very important in meiosis and in our germline, because that's what produces a unique human, right? If this crossing over did not occur, then we will most likely all look just like our parents, right? And so this is what gives us different allele frequencies in our species, right? In a species in general, actually. So this is a synapsis right here, and this is where crossing over occurs, right? And this is in prophase 1. I'm just gonna write CO as crossing over. And what this structure is called right here, when there are 2 homologous chromosomes are joined together, is called a bivalent, okay? So a bivalent, I'm sorry for the handwriting, by the way, but a bivalent is 2 homologous chromosomes or 4 chromatids, right? So as you can see right here, it explains it really well, right? So that's what goes down in early prophase 1. Now, also, we gotta remember our spindle is forming as well, right? So our centrosome is gonna start building up our spindles because we need that later on to split our chromosomes apart, right? So we got that in play. And this diagram is really helpful because you can think of the red as the maternal chromosomes and the blue as the paternal chromosomes, and it will help you just understand how this works better, right? Now, so we formed that, crossing over occurred, and as you can see, in crossing over occurred in prophase 1, and also the nuclear envelope dissolves as well, right? Now, we move on to the next step, which is metaphase 1, right? In metaphase 1, our chromosomes are still set as bivalents, right? These are homologous chromosomes joined together as bivalents. So if Dr. Maduro asked, in this case, how many bivalents do we have? Well, we have 3, okay? Because remember, bivalents include 2 homologous chromosomes joined together or 4 chromatids together, right? In this case, so we see that we have 1, 2, and 3, right? And as you can see, there's a mix of red and blue, right? So that means that crossing over occurred, and each chromosome has its own, basically its own specificity at this point. And so these spindles will attach to each chromosome and pull them apart, just like you see in mitosis, right? In anaphase, they start getting pulled apart, and we form 2 different cells, right? Now we're not done yet. This needs to go through a separation of these sister chromatids now, and that's what will happen in meiosis II. So we'll go over that right now. So meiosis II is essentially but mitosis. In this case, we won't see any crossing over or anything. It's just gonna be simple division, right? So these are our 2 cells that we generated from meiosis 1, right? So we got our first cell here and second cell here. And essentially the same thing is gonna happen, right? So in prophase 1, our spindle forms again, as you can see here. In metaphase, our chromosomes line up in the middle and the spindles begin to attach, right, to our chromosomes. And this middle thing is called a metaphase plate, by the way. And then anaphase will separate our sister chromatids, and they begin to move at the opposite ends, right? And now we have 4 distinct cells, right? And they're N. So remember, we first started off with 2N before even meiosis 1, and now we're at N. So we went from diploid to haploid. So if you're talking about germline cells, you can talk about this being an X chromosome, this having the X chromosome, this one having the Y chromosome, this one having a Y chromosome. And like, think of these as your sperm cells, and one of these will attach to the egg, right? And that will form a 2N zygote, right? So that's just an overview of mitosis and meiosis. It gets really complicated when you start dealing with the vocabulary and determining the stages at which it is diploid and haploid. So just keep reviewing this, and good luck. And even if you ever have any questions. So thank you.